So thanks a lot for the invitation. Very happy to be here today and looking forward to present our work on the generation of an in-depth tissue-resolved proteomics atlas of the non-human primate Macaca Fascicularis on the timsUltra-AIP. First of all, I would like to introduce you the animal I'm speaking about today, the Macaca Fascicularis. To put our work into context, in the year 2018, more than 75,000 non-human primates were used for biomedical research in the US only. And among these, the long-tailed macaque, Macaca fascicularis, was one of the most widely and most frequently used animal. Non-human primates are widely used in the investigational new drug development, and here not so much in early discovery, but rather in later stages, such as safety assessments or translatability questions. And why is that? First of all, they are the closest living relatives to human that we can use as animal models. Second, they have high neurological similarities to human and are therefore very well suited to be used for neurological studies. A fact that was very interesting also to me is that they actually do develop naturally heart diseases, and we can use them for studying cardiovascular and metabolic diseases. Then they also do share very similar reproductive biology to humans, so they do menstruate and have a menopause. And I think a fact that a lot of you know is that non-human primates do have a very similar immune response to human, and they are therefore very well suited for the development of vaccines. A last point I listed here is that they are also widely used for pathogenesis and viral disease research. So Macaca fascicularis is evolutionary very close to human, but what do I mean by that? On a DNA level, that means we share about 93% similarity, and on a transcript level, it is about 86%. But we all know that most of the diseases are actually happening on the protein level. So therefore, I was asking the question, what is about proteins? And here, I don't only refer to protein sequence similarity, but also to protein expression similarity. Although non-human primates have been widely used as animal models, there was no systematic approach done in order to create a large proteomics atlas or data set. And this is where we wanted to address the gap and create a tissue-resolved proteomics atlas for non-human primates. So we selected macaca and human and about 32 different tissues and did classical bottom-up proteomics sample preparation. We first did lysis followed by digestion and injected these peptides into an EvoSep timsUltra-AIP connection. We were using Spectronaut in the cloud for the data search. So to give you some more details, we had a total of 7 animals, so 7 macaques, whereas 4 were males and 3 females. For human, we had n equals 1, and this resulted in about 200 tissue samples. These were then measured with a whisper zoom 20 samples per day gradient using a DIA PASEF method on the timsUltra-AIP. And we used omics flow, so Spectronaut in the cloud. I would like to give you some more details on the second part of the workflow and start with the DIA PASEF method we used. So we did use an optimized method, and what do I mean by that is we pruned the space, so we did not use the entire M over set and also not the entire ion mobility space. And this to give the TIMS more time to spend for the relevant ion population. So first we did a NHP, we measured the NHP brain pool sample with the full range and identified 8,452 protein groups. And then we measured it with the optimized method, so with the pruned one, and reached 9,316 protein groups. And that is a stunning increase of 10% in protein group identifications, which is to me very phenomenal to see that by just spending more time on the relevant iron population, you can boost your ID so much. Now let's move on to the second part. I think a lot of you know that data coming from either an Astral or an AIP can be quite big, the raw file size, and therefore also the search can take quite a lot of time. And the potential solution to that is running your search in the cloud and therefore parallelizing your runs. So what we did is we searched 163 NHP tissue samples with SpectroNode on a local workstation, and this took us about 116 hours. We repeated the same search, but this time in a cloud, and we were done after not even a day, and therefore could reduce the overall time by 5-fold, which of course gives you back a lot of your time. But now let's jump right into the NHP dataset. Here we are looking at a bar plot with the NHP tissue samples on the x-axis and the identified protein groups on the y-axis. We were able to identify 2,470 protein groups in all of the samples and reached almost 8,800 protein groups in average. We were able to identify 13,846 protein groups cumulatively, and to our knowledge, this is the largest proteomic dataset created for the non-human primate Macaca Fascicularis to date. Now looking at the global proteome patterns, we actually see some tissue-specific clustering, and I would like to show you 3 examples what I mean by that. The first one being tonsil, spleen, lymph node, and thymus clustering together, and this is because of their shared immune-related biology. The second example is heart and aorta. They are anatomically and functionally related tissues clustering together, and this is because they share cardiovascular biology. The third example is brain and eye, and also here they are clustered together because of their shared neuroproteome signatures. And these patterns give us confidence that the dataset actually captures true tissue biology rather than technical artifacts. Now let's compare the NHP dataset to the human one. In the middle we have listed the shared tissue types, and on the left-hand side you see the dataset we saw before, but this time averaged per tissue type. On the right-hand side it is the human proteomic dataset. And we also here we can actually see some similarities across the species. For example, heart is among the lowest, among the lowest protein group identifications, and testis shows among the highest protein group identifications. And this mirrors observation from human tissue and is actually very, we can see conserved proteome signatures across these 2 species. So looking at the principal component analysis, it is getting visible that we see a clear species separation on the x-axis. So we have principal component 1 on the x-axis and PC2 on the y-axis, and each dot is representing a single sample. And it's really clear that we have a clear species separation. But what it's even nicer to see is when we change the coloring based by tissue type, we can see a tissue type clustering on PC2, or in other words, we can actually see the positioning of the different tissue types in the 2-dimensional space is highly similar between macaca and human. And this again highlights conserved proteome organization across species. Beyond global similarities, we also looked at evolutionary innovation. So going back in time, there was actually a separation of primates And to me, this is still very incredible to realize that we still share 93% of DNA similarity. But still, in that long period of time, of course, there happened some evolutionary changes in the genome. Looking into literature, I found a publication from Ruiz Orrera and colleagues from the year 2024, where they were looking at these evolutionary innovations, and they actually found that the innovation often occurs at the level of gene expression rather than the gene sequence. So what they did is they calculated the tissue specificity, here specifically the cardiac specificity, and found it to be higher in human genes than in the non-human primate. So further on, they were looking where the tissue enrichment is actually happening for these genes for the non-human primate, and it is in brain, cerebellum, liver, and kidney. But when checking these same genes in human, the tissue enrichment is happening in heart. So the authors concluded that this is a switch in the tissue enrichment. So we were actually curious how it is in our data set on the proteomics level. So we did calculate the same tissue enrichment and found 255 cardiac enriched proteins in human that is visible on this heat map. Now zooming in, it is also getting visible that there is a quite high overlap of a third of these proteins that are also tissue enriched in the non-human primate. So we can clearly conclude that we have conserved proteome architecture, so high similarity, but we also see differences, so protein expression is changing. We were then asking the question whether these proteins do relate to a disease and did disease ontology enrichment analysis. And we found actually that these 255 cardiac enriched proteins in human are overrepresented for cardiomyopathy-related disease. So we can see that these proteins we found are not just random, but they can be correlated to a cardiac disease or even cardiac disorder. So taken together, what we just saw is we could, as a first, we could confirm the trend that was visible on a gene or transcript level, also on the protein level. But it is also important to realize, so overall we have a high conserved proteome architecture between these two species, but we also have similarity, which is very important when asking translatability or safety questions in drug development. But let me go back to the initial question of similarity. I now also talked a lot about differences, but what is now about this similarity, I asked in the beginning. So we did a group-wise Pearson correlation analysis across proteins stratified by tissue and sex. And here each point is reflecting a tissue proteome, and we see that the overall correlation between macaca and human is high, with a Pearson correlation of 0.8. And you were probably wondering what this point down here is, and it is actually eye. And you can probably imagine it is not that easy to lyse a full human eye, so therefore we have a slight difference here. But I would like to focus on the safety-relevant tissues such as heart and liver. So here, looking at the, here we see each gray dot is representing a protein, and we can actually appreciate that the overall correlation is very high, again with a Pearson correlation of 0.81. We were then interested in looking at some specific heart biomarkers, and also these align pretty nicely between macaca and human. Another example is liver. Here the Pearson correlation is even slightly higher, and looking at some specific liver markers such as these CYP proteins, which are important for metabolic processes, are also very well aligned in the middle between macaca and human. So overall, we could really see that there is a high similarity of proteome architecture between non-human species and human, but we still see some similarities and differences. So let me summarize what you just heard. We were able to create one of the deepest proteomic data sets for the non-human primate Macaca Fascicularis. Further, we were able to show tissue-specific clustering, which is actually biologically meaningful. We saw some similarities and differences, which are very important when you ask safety or translatability questions in drug development. Further, we hope that with this data set, we can support the 3R principle, which is replace, reduce, and refine of animal models. And together with that, we also hope to improve translatability across species. For all the proteomics people in here with the created peptides, we also hope to support and create targeted assay development. And as you can imagine, with the data set, you can do much, much more, and there are basically no limits. And with that, I'm almost at the end of my presentation. I would like to thank the bunch of people that was involved in this huge project.